Good morning, everyone. Thanks a lot for being here. Thank you, Deepa, so much for inviting me to speak here today. A key theme of the synthesis era going back to Fisher was the rejection of non-Darwinian theories that were held to be incompatible with genetics or population genetics, including the idea that evolution might exhibit tendencies or trends that are due to internal tendencies of variation. So, for instance, when Simpson reconciled the fossil record with this synthesis, he looked at potential trends. He argued, well, they're probably not real trends, and he concluded that even if they are real trends, they must be due to selection, because selection's the only known cause of direction, and the only alternative is vital urges or cosmic goals without known mechanism. Now, where did Simpson get this confidence to just reject an entire class of theories? Why does he say there's no mechanism when previous thinkers had simply assumed that tendencies of variation would cause evolutionary tendencies? Well, we know the answer to this question. Simpson and the architects of the modern synthesis did not reject the possibility of tendencies of variation. They rejected their efficacy as causes of tendencies in evolution. Fisher and Haldane argued explicitly that mutation is a weak pressure. Because mutation rates are so small, mutation is a weak pressure, unable to influence the course of evolution. It won't be important unless mutation rates are abnormally large or selection is absent. This argument was repeated by the architects of the modern synthesis and became part of that 20th century orthodoxy, and it's still with us in various ways. So in the literature of molecular evolution, for instance, you still see the assumption that mutational explanations for patterns require or imply neutrality. Another example would be Hirsi Gould in his 2002 book. He cites Fisher's version of this opposing pressures argument and concludes that since orthogenesis can only operate when mutation pressure becomes high enough to act as an agent of change, empirical data on low mutation rates sound the death knell of internalism. So what does that mean for the idea of developmental constraints in evo-devo? How would developmental biases influence evolution? Well, it's not clear. The issue hasn't been resolved. If you go back to the famous 1985 paper by Maynard Smith and colleagues that defined this issue of developmental constraints, they bring up the Haldane-Fisher argument. They say that it deserves re-examination, but they don't provide a solution. So today, we still have population geneticists who think that evo-devo is illegitimate, and it's the default view now in evo-devo that population genetics does not provide a sufficient account of causation to understand the role of development in evolution. But the problem is that the Haldane-Fisher argument is mistaken. It assumes standing variation. If you have a process of evolution that depends on events of mutation that introduce new alleles, that process will be subject to biases in introduction by a kind of first-come, first-served logic that Haldane and Fisher didn't consider. This kind of causation was demonstrated in a simple model of one-step adaptation with two choices in 2001. So imagine we've got a bunch of different possibilities. There's two that are advantageous. We can go to the right with a slightly higher selection coefficient but a lower mutation rate, or to the left with the opposite of that. And the question is, how often is evolution, and under what conditions, is evolution going to go to this mutationally favored option? So here are some simulations showing the bias in outcomes as a function of mutation bias for a bunch of different population sizes that entail going from small mutation supply to large mutation supply. And what we see is that all the lines are going up, and wherever the bias in outcomes exceeds 1, that means that evolution is favoring the mutationally favored option. Now, as I said, this wasn't part of the view of Haldane and Fisher because they assumed evolution from standing variation. If all the alleles that are relevant to evolution are present in the initial population, mutation just shifts the frequencies of things that are already there, and it's not very important because mutation rates are so small, just like Haldane and Fisher said. And we can show this using the same model. All we have to do is just put those alternative alleles into the initial population. So that's what I've shown here. So this upper curve here is just evolution from new mutations, like I showed you before. The lower curves here are where we've put the alternative alleles into the initial population, into the gene pool of the starting population. And the result is there's almost no effective mutation bias. All right? And mutation is still happening under these conditions in these lower lines here. Mutation is still biased. It just, none of that matters if there's no introduction process. So that's why when I refer to this theory, I refer to biases in introduction. It's not the same thing as mutation bias. You can have mutation bias and mutation without introduction bias. And even if all mutations happen at the same rate, if you're considering phenotypes, the asymmetric mapping of phenotypes to genotypes can induce biases in the introduction of phenotypes, of alternative phenotypes, even if there's no mutation bias. So you can have mutation bias without introduction bias. You can have introduction bias without mutation bias. They're not the same thing. Okay, so what do we know about this kind of causation? So there's theoretical work on this that comes out of 5 different groups. Recently, Suarez et al. Did a full analytical treatment of this one-step adaptation with 2 choices. There's this great model of 2 competing traits from Joanna Mausel's lab, that's Gomez and colleagues. This spring, Kano et al. Came out with a paper with a lot of simulations of a gene model and this really interesting way of capturing the total influence of mutation with this regression parameter beta that ranges from 0, no influence, to 1, proportional influence. And several groups have done modeling of adaptive walks on complex landscapes under a bias in mutation or a bias in introduction. And I'm particularly interested to draw attention to this work from Ard Lewis's lab on the findability of RNA folds. Well, I don't have time to go into that. So what these modeling studies tell us is, first of all, the factors that matter in the efficacy of biases in the introduction process. So mutation supply is really important. Strength of selection, the diversity and size of the target, that is the part of the distribution of fitness effects that's beneficial when we're talking about adaptation. And then they document some interesting outcomes, like this degree of mutational influence can actually range from 0 to 1, depending on population genetic conditions. Other studies have shown systematic compositional trends, differences in landscape navigability, and the findability of intrinsically likely structures. So we have at this point, and the broader context, just in the development of evolutionary thinking, is that this theoretical work gives us a warrant to draw a kind of arrow of this green arrow of causation here between tendencies of variation and tendencies of evolution. That's the arrow that Simpson and the architects of the modern synthesis thought was incompatible with population genetics. And that means in a broader context, we can consider mutational and developmental causes of tendencies and variation and link those potentially to interesting patterns in evolution, like parallelism, self-organization, and so on. So what evidence do we have for this kind of causation? The best evidence for sort of direct demonstration of causation would be laboratory experiments. So on the left here, I'm showing the relationship of frequency evolved in replica cultures of rifampin-resistant variants as a function of their mutation rate, data from McLean and colleagues, showing this strong correlation. And on the right, we have a study of cefotaxime resistance under different mutation spectra from Cousset and colleagues. So this plot is a little bit hard to read. The This is showing the numbers of strains that achieved resistance in different ways. The blue strains they're colored by their parent strain. So blue comes from the mut T parent, red comes from the mut H parent, black comes from the wild type parent. And then they're put in boxes according to what type of mutation it is. So the reason all the mut T strains are in this box on the left is that's the box that has the type of mutation that's favored by mut T, which is AT to CG transversions. The middle box is transitions. Those are the ones favored by mut H, so that's where all the red strains end up. And then wild type strains can adapt by various types of mutations, and they're spread in those 3 different boxes. So that shows this kind of effect of mutational introduction or mutational origin on adaptation in the laboratory. And it's very direct, and you can't explain it with the mutation pressure theory because it doesn't require high mutation rates. It doesn't require neutral evolution. It doesn't require absolute constraints between possible and impossible variations. It's an effect of quantitative biases in introduction. What evidence do we have for this in nature? Well, in recent years, it's become possible to, we have enough detailed information now to do statistical tests for effects of mutation on these famous cases of adaptation traced to the molecular level. So, for instance, at the top here, spectral tuning. At the bottom, hemoglobin adaptation in high-altitude birds, tetrodotoxin resistance, various other cases. A few years ago, we collected data on 10 of these cases covering diverse taxa and various proteins, and we looked for an effect of transition-transversion bias. So the way to read this pass and events distinction. So, for instance, in the second row, tetrodotoxin resistance, there's 8 different ways that this has evolved in parable, eight different amino acid changes. 3 of them are transitions and 5 of them are transversion paths, and the 3 transitions happen 2, 6, and 3 times, and the 5 transversions happen 2, 2, 2, 3, and 3 times. So each of these numbers of events is 2 or more because we're only counting parallel changes. And if you add up whether by paths or by events, we see a strong significant excess relative to the null expectation of 1 to 2. And so that's evidence that ordinary biases in ordinary types of mutation influence what happens in adaptation. I don't have time to talk about this today, although, again, I want to draw your attention to this work from Art Lewis's lab on findability of RNA folds. So just to summarize, we have evidence that falls in the category of microevolution, looking within populations over short periods of time. Sources that are mixed. So the study that I just showed you, that meta-analysis of transition-transversion bias, some of those were sort of recent local adaptation. Other ones were like spectral tuning that happened millions of years ago. And then there's these studies of long-term innovation from Joanna Mausel's lab and Art Lewis's lab. And notice that all of this is quite new. So just like the theory that I showed you, most of this is from the past 5 years. So because of this, we're constantly seeing new things that we didn't think about before. And I'm going to tell you about one of those now that has to do with the conservative transitions hypothesis that, again, came as a surprise to me and even a little bit of an embarrassment because of something I'd written about this. So in protein sequence evolution, transitions happen more often by transversions, and that might be due to a mutation bias. But a long-standing idea in molecular evolution is that transitions are more conservative at the level of proteins. And this conservative transitions hypothesis has been around for a long time without being directly tested, but now we have high-throughput fitness measurements. So we can just ask, are transitions more conservative? So we gathered data from, this is data from about 11,000 fitness measurements from deep mutational scanning studies, and they're aggregated together by fitness quantile. And so on the left, you see the 12 different nucleotide changes, and there's some subtle differences in the fitness distributions there. But when we aggregate them into transitions and transversions, there's basically no difference. All right? Transitions are not more conservative than transversions at the protein level. That's what high-throughput fitness data say. Now, we were also interested, but this is covering the whole fitness distribution, everything from 0, which is not very, the low part of the distribution is not very important. We were curious about what's happening at the high end of the distribution, so we looked at some adaptation experiments where fitnesses were measured. And we did rank order statistics where we're just asking are transitions better than transversions among these adaptive changes? And we asked that by saying, what's the chance that a randomly chosen transition will be more fit than a randomly chosen transversion? And if we're drawing from the same pile, it's going to be 50%. That's the null expectation. So here's 4 small studies, and this statistic, what's the chance that a transition is better than a transversion? And in every case, it was less than 0.5. And in the aggregated data here, it was 0.40, and it was on the margin of significance here. And when I saw this result, I just laughed and I said, ha ha, not only are transitions not more conservative, transversions are actually better when you look at beneficial changes. But then when I started to think about it more, I realized that's not a safe inference, because these sets of changes that we're looking at here, our observation of these changes is conditioned on them having on the degree of mutation rate and on the degree of fitness benefit. And when you condition on a joint distribution like that, you can get, that can induce non-causal associations, and it can result in paradoxes like BERKSON'S paradox. So what's BERKSON'S paradox? So suppose we have a distribution of talent and looks in a population, and they're completely uncorrelated with each other. But we're curious about celebrities, and suppose that celebrities become celebrities by having a high sum of talent and looks, okay? Let's say it's the top 5% of the sum of talent and looks. Well, what that means actually is that in this celebrity population, there's going to be a negative correlation between talent and looks, because the celebrities that happen to achieve celebrity status in spite of modest looks must have been very talented, and vice versa. And this might be what's going on, maybe with transitions and transversions. Maybe the transversions that we see in adaptation, in spite of having a slightly lower mutation rate, tend to have a slightly higher fitness benefit. So here's an example that I saw recently. This is just from a preprint that came out about 6 weeks ago from Watson and Blundell. This is about clonal hematopoiesis. And I just took their figure there and I replotted it to show that there's a negative correlation between selection intensity and mutation rate, all right, for data on clonal hematopoiesis. But we don't know that that's a negative association induced by conditioning, because we don't have the prior distribution. We don't know what that looked like. And in general, we want to look at this problem in a more disciplined way. We need to have some theory for this. What would that look like? Well, in general, there's some joint distribution, okay, of selection coefficients and mutation rates, and it's going to change from a prior distribution to the distribution after mutation to the distribution after some selective process. So here's a possible distribution over here of selection coefficients and mutation rates, and the distribution of possibilities that doesn't have any weights on it, I'm going to call the nominal distribution today. All right? So all these dots are the same size. Mutagenesis turns the nominal distribution into a de novo distribution. Okay? That's what mutation looks like. So to get that de novo, we're just multiplying by the x-axis here. So things are going to get more dense going to the right along the x-axis. For fixation, then, we're changing this distribution by the vertical axis. So it's the weights are going to go up vertically. All right? So this is the kind of change that we're going to see. And we've done some mathematical theory on this, on what do we expect to happen to these distributions, a nominal distribution, under mutation, and then some kind of selective process. And the answer is it's complicated. Lots of different things can happen. So the effect of conditioning on the covariance of selection coefficients and mutation rates, it can be negative, so it can be like BERKSON'S association, or it can be positive. It depends on the shape of the nominal, and it depends on how selective filtering happens in a population. So what this means is that rather than just relying on abstract theory, we need to look at some actual distributions. So the distribution that I just showed you a moment ago is an actual distribution. So this is based on a deep sequencing study of dengue virus grown in human cell culture. And what we have here are data on 325 beneficial mutations in the whole genome. So we have measured fitnesses for these. We don't have all the measured mutation rates. Instead, there's just a mutational model, a context model for mutation. And so for the de novo here, we're multiplying this by the mutation rate model. And for the fixed distribution, we're simulating a process of evolution, okay? And what happens is that the correlation goes from being slightly negative to being very negative to being even more negative in the fixed distribution. And this, as I mentioned, this can be affected by how population sampling happens. So this is actually for a mutation-limited regime, where it's just the probability of fixation. We've also simulated this under different conditions. So these are the same 3 plots I showed you before, but here's 2 more fixed distributions down here where the mutation supply is different. So here it's 100-fold higher, and here it's again 100-fold higher. So the correlation gets less negative and even less negative here, and the general effect is shown on this plot. So at very low mutation supply, we get the most negative correlation, and then it kind of goes up as mutation supply increases. So that's interesting to look at, but really we don't want to rely on simulations. We'd like to have some data where we have both the nominal distribution and we have the fixed distribution. And that's really difficult to get. We don't have any really great data on that for evolution, but we can get something like it when we look at cancer. So I'm going to show you these results for cancer before I wrap up. So we have a nominal distribution of fitnesses that's from deep mutational scanning of P53, and it's important to remember that cancer driver mutations, they're typically breaking P53. So there's lots of beneficial mutations. There's like 8,000 amino acid changes, 2,000 of them are beneficial according to this kind of assay. And so it's beneficial from the perspective of the cancer, not from you. The mutation rate model is a mutational signature model like they use in cancer studies all the time, and the prevalence data is going to come for the fixed distribution is going to come from the Gini database. So what we have here are those 3 distributions again. So in the nominal, there's a slight negative correlation, and in the de novo, just very, very slightly positive. In the fixed distribution, it actually becomes a much more strongly positive. And this is again something that's affected by, or at least we think it's something that's affected by the way selection is happening. So we've tried to simulate this. So here we're simulating we're using that nominal distribution and the mutation rate model, and then simulating selection for these different cancer drivers. And the conditions under which we see the highest correlation between our simulated results and the observed results is at intermediate mutation supply, and likewise, the correlation between mutation rates and selection coefficients gets most positive under intermediate mutation supply. Okay, so these are results for single nucleotide mutations. We have a model for single nucleotide mutation rates that we're using here, but the prevalence data and the deep mutational scanning data also have multinucleotide mutations, and that raises an interesting question. So, well, we don't have a good rate model for multinucleotide mutations, but we know that they happen typically. They happen 2 or 3 orders of magnitude less frequently than single nucleotide mutations. So we can just compare those 2 categories, and the question is, do these rare types of mutations tend to have stronger benefits in order for us to observe them as cancer drivers? So here's this result in the nominal distribution. Okay, this is just from deep mutational scanning. Of course, there's a large number of multinucleotide changes in these deep mutational scanning studies. And in this one, and again, remember, these are mutations that break P53, so there's lots of beneficial ones in both categories, the singles and the multiples. And by this statistic of comparing from one category to another category, the chance that a multinucleotide mutation has a higher fitness is about 0.61, so they're like modestly better, but a very small effect. But here's what happens when we look at the fixed distribution based on cancer prevalence. So there's far, far fewer doubles or multiples relative to this, and that's because the mutations are really, really rare. But when we see them, they tend to have a higher fitness benefit. They're better drivers. And that's shown statistically here, so the chance that one of the multinucleotide changes has a higher fitness, according to the deep mutational scanning estimate of fitness, is 0.88, and that's a significant result. Okay, so I need to wrap up here. This has been an exciting project. It's been going on for just a few months. The stuff that I'm showing you here is very raw. And we need better data sets, more fit to purpose data sets. It's been a pleasure to work with the team that I'm working with on this. So Brian Gitschlag and David McCandlish are responsible for the theoretical work, most of which I didn't show you. Alejandro Cano from Josh Payne's group in Zurich did all the empirical analysis and simulations. I probably don't have time to talk very much about the big picture. No, I don't. I haven't heard the gong yet, but it's been 24 minutes. Anyway, so I will just stop here. I'd be happy to talk about these bigger questions about like where we are right now, where we're going, where we have been. For instance, this idea of mutation-biased adaptation went from being a heresy for the people who cared about it to being controversial for the people who cared about it, to being something that for the people who care about this, we consider this to be well established now, although I'm not sure what anybody else thinks. So I'll just wrap up there, remind people that you can always contact me online if you have questions about this that only come to you later. I'll also look at the chat, the meeting chat, and try to answer any questions there. Thank you very much. So the question is whether the model accounts for multiple cell lineages. I'm not sure I understand it, but we're sort of mashing together the data that we have about cancer, and again, the prevalence data are just the prevalence that you'd get from the Gini database for cancer drivers. It's aggregating across different types of cancers, all right, but they're all united by having p53 drivers, right? Right. So so the question is, what if there's multiple drivers that are contributing to the cancer? Yeah, and we're just not getting into that level of sophistication. We're just trusting whatever we get from the Gini database. But if you have some insight on that, maybe you could talk to me afterwards. Because the cancer data is new to us. We're mostly looking at evolution. Go ahead. Well, when you're conditioning on repeated occurrence, you're going to enrich both for a higher fitness benefit and a higher mutation benefit. Now, you could be enriching for one more than the other based on some complicated feature of the background distribution, right? And I don't know the answer to that. But that's not vitiating the test that we did, because remember, there's a test for paths and there's a test for events, and the test for paths doesn't take into account. So the reason that we conditioned on parallelism is just we considered that to be further evidence that these are actually real adaptive changes, because that's the big problem in molecular evolution. When you look at these changes that are candidate adaptive changes, are they really adaptive changes? And in that study, we demanded they have to occur like in two different species, and there has to be a functional result, like like genetic engineering, showing that this change has a functional effect that's consistent with the adaptive story. So if the adaptive story is this increases hemoglobin affinity in high-altitude birds, then you have to engineer that change and show that it actually increases affinity. So those are the criteria for inclusion in that study. Does that answer your question? Okay. Anything else? All right, thank you very much.